introduce Dr. Bob Data to present to us as part of the Wednesday afternoon Director's Lecture Series. Bob originally comes from Long Beach, California, but then completed much of his training in the Northeast. He received a BS in molecular biophysics and history from Yale prior to matriculating to Harvard Medical School, where he completed his MD degree as well as a PhD in neurobiology. Following that, he relocated to Columbia University, where he was able to work with the Nobel laureate Richard Axel. Bob, I assume right around the same time that you were training. So an interesting time to be a part of the lab. Following that, he then joined Harvard as faculty. This was in 2009, and proceeded through the ranks quite quickly, reaching the rank of professor just 12 years later in 2021. Bob is a scientific advisor, member of advisory boards throughout the country and world. He supports industry, academia, as well as here at home, where he's an advisory board member of our National Smell and Taste Center. Bob has contributed to peer review, the NIH, NSF, multiple other places, and is truly a resource, a leading voice within the olfaction and chemosensation communities. Bob studies how the brain organizes and integrates information about sensation to drive meaningful interactions, how we take signals from the world around us and integrate that into brain function and interactions. This is spanned from detection to CNS integration all the way forward to action. And I'm incredibly excited to hear some of his newer findings and work that I believe has the potential to truly push our field forward. So with that, and no pressure, Bob. Thank you. I had two tasks, and Bob, just 1 second. For everyone joining us, if you would like to receive CME credit for this event, please enter the code 57691 in the Johns Hopkins CME portal. Thank you.
Great. Thanks so much, Josh, for that really kind introduction. And thanks to all of you here at the NIH. This is really an honor for me and for my lab. This is the nation's preeminent biomedical institute, and so I'm truly grateful for this opportunity to catch up with old friends, to be with all of you, and to share some of our science. So as Josh mentioned, my lab's main interest is in thinking about how the brain takes in sensory information and then generates natural behaviors out in the real world. And today I'm not going to talk at all about the work that my lab does, trying to understand the neural basis of natural behavior. Instead, I want to talk about some work that we've been doing, exploring the sense that is most important for most animals out in the real world. And that is, of course, the sense of smell. So smell is elusive, right? We think of smell as being kind of primal in animals. It lets them smell food predators and mates. It lets them survive and thrive in the world. But for humans, we think of smell as being kind of aesthetic, although, of course, smells are capable of eliciting deep memories and emotions. And perhaps it's because of the close connection between smell and emotion that people who acutely lose their sense of smell often feel bereft and are at increased risk of psychiatric disorders like depression. And maybe because of the close relationship between smell and memory, people who have new onset smell loss are at increased risk of neurodegeneration. And so it's important that we understand smell, not just because it helps us to understand how the brain makes sense of the world, but also, of course, it's important for human health and wellness. So today I'm going to tell you a bunch about the canonical model for smell and why we think a major part of it is wrong. But before I do that, I want to talk a little bit about maps. So what is a map? A map is a symbolic representation that captures some feature about the world and which maintains relationships. And so a great example of a map that I think should make what I mean clear is, of course, this map of the United States. So here, the feature that's being captured is elevation, and the relationships that are being maintained are spatial. And so armed with this kind of map, you can know how high a city is, and you can make predictions about how hard it's going to be to bicycle from point A to point B. Maps don't just have to capture physical features about the world. They obviously can capture more abstract things like, in this case, political boundaries, right, as you see here. And one of the great things about maps is that they're easily reproduced and transformed. As you can see here, in this famous painting called Map by Jasper Johns, which here is meant to emphasize the porosity of the political borders that separate the states and to remind us that we all belong to the same country. Now, maps are pervasive in the nervous system, and the maps that we understand the best create internal representations of the external world for our senses. And we particularly understand those for touch, for sight, and for hearing. So, for example, we know that the retina contains within it a retinotopic map of the visual world, such that neurons in the retina that are near each other tend to represent objects that are near each other in visual space. Similarly, the cochlea contains within it a so-called tonotopic map, such that neurons that are near each other in this physical space tend to respond to sounds of similar frequencies. And so mapped out in physical space are key sensory characteristics in these retinotopic and tonotopic maps. And importantly, the structure of these maps are maintained as information progresses from these peripheral sense organs into the thalamus and up into the cortex, which itself contains within it retinotopic and tonotopic maps. We know a lot from development about how these maps are built and how they're propagated from one brain area to the next. And a main theme is that gradients, chemical gradients, play an important role in the development and maintenance of these maps. So in the case of the visual system, we know that the retina contains a gradient of a molecule called Fa, and the thalamus, its target, contains a gradient of a molecule called ephrin A. These gradients tell neurons in each of these areas where the neurons are in space, and they help guide axons in a point-to-point manner, allowing this retinotopic map to be maintained in this higher brain region. And I'm telling you this because the idea of gradients is going to come back to us later in this talk. Okay, so what do we know about smell, and how is smell mapped in the olfactory system? So smells are, of course, detected by odor receptors, which are 7 transmembrane G protein-coupled receptors, each one of which is specialized to interact with a subset of odors outside in the world. There's a ton of these receptors in the genome. In the mouse, there's about 1,000 odor receptors. In the human, there's about 400 odor receptors. These odor receptors are expressed in the cilia of sensory neurons, olfactory sensory neurons, that live here in the olfactory epithelium within your nose. And it turns out through molecular mechanisms that we don't fully understand that mature olfactory sensory neurons choose to express one and only one of the, say, 1,000 possible odorant receptors that can be expressed in the mouse. And so what that means is that there are 1,000 different olfactory sensory neuron subtypes, each one of which is defined by which receptor it expresses. So we have 1,000 different types of receptors. How are these receptors organized in space in the epithelium? I told you there are maps in the other senses. Is there a sensory map here in the nose? So what you're looking at here on the right is a whole mount of a mouse that's been genetically modified such that all those neurons that express the same odor receptor, so one channel out of 1,000, express the marker GFP. And what you can see is that these neurons are actually arrayed along a stripe, which in the field is referred to as a zone. And the zone traverses the dorsiventral extent of the epithelium and occupies about 1/4 of the epithelium. And so it's thought that there are 4 of these so-called zones. Okay? And each odor receptor is somehow thought during development to be assigned in a stereotyped manner across animals to one of these 4 zones, and therefore to live in one of these 4 stripes. However, within each zone, it's thought that basically cells pick randomly from one out of about 250 receptors. And so to the extent that there is a map in the nose, that map is composed of these kind of 4 very vague, very broad zones. And the vibe of the nose is that basically neurons develop when they randomly pick a receptor. Okay? It's important to note that this process of odor receptor choice doesn't just happen during development. It also happens continuously throughout the lifespan. So just underneath a layer of mature olfactory sensory neurons, there exists this layer of stem cells that's continuously spitting out neural precursors and intermediates and ultimately generating a whole new set of mature olfactory sensory neurons that have to choose new receptors. And these new neurons are continuously being integrated into this circuit. Okay, so how does information get from the nose to the brain? Well, all those neurons, all those neurons that express the same receptor elaborate axons that puncture this bony cribriform plate at the base of the skull, and they all converge on a single insular structure within the olfactory bulb in the basal forebrain, and the structure is called a glomerulus. There are 1,000 different receptor types. There are 1,000 different types of receptor in the nose, and so there are 1,000 different glomeruli here in the olfactory bulb. And it turns out that the spatial position of each one of these glomeruli, as determined by which receptor innervates it, is invariant from mouse to mouse to mouse, and human to human to human. And that has a very important consequence. Different odors are going to activate different receptors, and therefore different sets of neurons in the nose. And that's going to go on to activate different spatial patterns of glomeruli in the olfactory bulb. So here's some functional imaging from my lab. You can see the spatial pattern of glomerular activation for jasmine is different from that of rose, is different from that of fox. And critically, because the spatial pattern of glomeruli is invariant across animals, through computational methods, I can just look down at one of these patterns and tell you what smell the mouse is smelling at any given moment in time. And so there's an incredibly precise map of odor identity built by the precision localization of glomeruli within the olfactory bulb. Where does information go from here? It's sent to multiple higher brain regions responsible for odor identification and odor and memory and emotion. And somehow these areas collaborate in order to build odor perception. Okay, so what have I told you? I've told you that basically, unlike the other senses, the nose barely has a map. Right? There are these 4 weird zones, but basically each neuron in the olfactory epithelium randomly chooses which receptor to express. And so the nose is disordered. Somehow, through mechanisms we still don't understand, axons from the nose get to the bulb where they build an incredibly precise map of glomerular identity, which organizes information about odor identity in space, as I showed you. And somehow the brain reads that code out to generate perception. Okay? So I want to talk today about this idea of disorder and these zones in the nose. And so I just want to explain to you a little bit about these zones. Okay, so this is the image I showed you before. This is a hole mount. This is a cartoon of a hole mount kind of depicting the zones from dorsal to ventral. Now, in olfactory science, we tend not to do these hole mounts. Instead, we tend to cut coronal sections through the olfactory epithelium, which reveal these kind of complicated turbinates that you all know about. And the important thing I want you to remember is that this dorsal to ventral anatomy maps into this coronal section such that dorsal in this view is actually kind of medial or proximal here in the epithelium, and ventral in this view is kind of lateral or distal. Okay, so this stuff in here is medial or dorsal, and this stuff out here is lateral or ventral. And so this ventral olfactory sensory neuron, which lives in the ventral, one of these ventral zones, if you look at that exact receptor in coronal section, you see it's kind of on the outside, as shown here. So that's just to orient you to the data that's coming. All right? So a question you might ask is, how are these zones, shown here, in the epithelium related to where olfactory sensory neurons project their axons in the bulb? So it turns out, as I mentioned, there's these 4 zones in the epithelium running medial to distal, 1, 2, 3, 4, and they roughly correspond to 4 zones in the bulb running dorsal to ventral. And so we think there's some mapping from here to here, kind of very crudely. Okay? And we know a little bit about the molecular mechanisms that support that. We know there's a marker protein that specifically marks the dorsal domain of the olfactory bulb that I'll tell you about in a minute. And we know there's a couple of axon guidance genes that seem like they might play a role in converting this zonal organization in the epithelium to an appropriate dorsiventral localization of glomeruli in the bulb. But I want to remind you that the olfactory bulb is incredibly precise. Right? All 1,000 glomeruli are very precisely located. And so this possible zone-to-zone mechanism of axon guidance cannot explain how it is that we have such a precise code for odor identity in the bulb. Okay, so now let's start talking about science instead of background. So the question we wanted to ask is, could we identify somehow molecular markers that identify all the different zones in the nose? So there's 4 zones, and we wanted to know whether we couldn't identify a marker for zone 2 or a marker for zone 3. Reasoning that if we could identify such a marker, we would be able to understand how odorant receptors are somehow limited to one of these 4 zones. Because that was our goal. How did we do this? We did what you'd expect us to do. We did a ton of single-cell sequencing. So we took the epithelium out of a mouse nose, popped it out, dissociated it, used standard microfluidic single-cell methods to develop transcriptomes for each cell from the nose. Okay? And what you're looking at here is a UMAP plot where each dot is an individual cell. And I'll remind you that proximity between points in this kind of plot represents similarity in their transcriptomes or the genes that they express. We get both precursors and mature cells. And here in this plot, mature cells are kind of highlighted by the marker OMP, which is shown here in yellow. And I just want to emphasize a couple points about these data. First, in order to do the experiments I'm about to describe, we've generated the largest single-cell sequencing data set ever assembled for any neural tissue. The data set I'm going to describe has about 5.5 million total integrated cells in it, including 2.5 million mature olfactory sensory neurons, and as I'll show you, about 400,000 precursors. The second thing I just want to mention is that, of course, analyzing data at scale like this requires sophisticated computational methods. I promise not to show you any math at all, but just understand there's a big engine that kind of underlies this. And using that engine, we very carefully searched for genes that might mark these different zones. And we found none. Okay? So the idea that there's some molecular code specifying these zones, even though we believe these zones are real, or had believed, turns out to be wrong. Okay, so we then decided to ask a much harder question. The question we decided to ask was, can we identify genes that systematically vary across olfactory sensory neuron subtypes? So we took all of our cells and we binned them. So we found 1,000 cells that express ORA, 1,000 cells that express ORB. We built 1,000 such bins. And then we just asked, can we identify any highly variable genes that vary across the olfactory sensory neuron subtypes? And the canonical view in olfaction is that every sensory neuron is basically the same, except for the receptor that it expresses, and so we expected to find nothing. Instead, we were able to identify 1,200 highly variable genes that distinguish olfactory sensory neuron subtypes. And this variation is such that if you just tell me 100 genes, 100 of these highly variable genes and how much they're expressed, I can with 100% accuracy tell you which odor receptors also expressed in that cell. Okay, so there's a transcriptional code that corresponds to OSN's subtype. We reasoned that these 1,200 genes probably were organized into coherent gene expression programs, groups of genes that co-vary in their expression up or down. And we used a mathematical technique called consensus non-negative matrix factorization to identify 7 such programs. And today I'm only going to focus on 2 of them, which are shown here. This, again, is a UMAP plot. Each dot, again, is a mature olfactory sensory neuron. And here I'm going to take the 150 genes that make up each of these JEPs. There's 150 genes that belong to JEP dorsal, and there's 150 or so genes that belong to JEP ventral. And I'm going to summarize the overall use of all 150 genes with a single number, and I'm going to heat map that over each cell. Okay, and I'm going to call that JEP usage. So what you can see is that some cells use JEP dorsal, and some cells use JEP ventral, but no cells use both. Okay? You might ask, why did we name these JEP dorsal and JEP ventral? We named this JEP dorsal because one of the 150 genes in this JEP was that marker gene that I'll tell you about for the dorsal domain. So we thought maybe this JEP represents something about being in the dorsal part of the epithelium. And we knew some of those axon guidance genes that seem to help guide axons from the nose to the bulb. We know that a couple of them are expressed ventrally, and those turn out to be part of the 150 JEP ventral genes. And so we kind of provisionally named these JEPs, JEP dorsal and JEP ventral, based on the hypothesis that these gene expression programs might convey some information about whether a neuron resides in the dorsal or the ventral part of the olfactory epithelium. Okay, we wanted to look at these JEPs a little bit more. And so we decided to look at them at the level of olfactory sensory neuron subtypes. So here, each dot is an olfactory sensory neuron subtype, meaning all those neurons that express the same receptor. And so there's 1,000 dots in this plot. And you see each olfactory sensory neuron either expresses JEP dorsal or JEP ventral, but not both generally. And if you express JEP ventral, you can kind of use it more or less. If you express JEP dorsal, you can kind of use it more or less. And the complementarity of this expression pattern means that we can effectively summarize the use of all 300 genes that belong to JEP dorsal and JEP ventral by just computing the usage of JEP dorsal and JEP ventral and subtracting them, and thereby computing what we call a dorsiventral score, or a DV score, which is how I'm going to refer to it for the rest of this talk. Cells that have low DV scores use JEP ventral a lot. Cells that have high DV scores use JEP dorsal a lot. Okay, so for each olfactory sensory neuron subtype, what is its pattern of usage of the DV score, which, again, is summarizing these 300 genes? What we see is that each olfactory sensory neuron subtype is associated with a unique mean DV score. So each olfactory sensory neuron subtype is using this set of 300 genes in a unique way. Here you see just some examples and their distributions, and here you see all of the olfactory sensory neuron subtypes ordered by their mean DV score. And you see that each subtype has a distribution, right? It's not each subtype, every cell that belongs to each subtype doesn't have exactly the same DV score, but they all have a unique mean. And as a consequence, this plot kind of tiles the possible DV scores. Okay, what do the individual genes that make up the DV score actually look like, and what are they? So here on the y-axis, you're looking at the 300 DV genes. And on the x-axis, we're going to look at individual OSN subtypes, and we're going to order them based upon whether they have low or high DV scores. And what you see quite clearly is that all of the DV genes that we identify via this method seem to be organized into gradients, right? Where there's a kind of gradient of expression of JEP ventral and JEP dorsal that varies depending on what your cell's DV score is. And interestingly, if we look at the genes that make up these JEPs, we find that they include a ton of genes that are about the function of olfactory sensory neurons, and two particular types of genes that we were really interested in. And those include transcription factors and axon guidance genes. And we were interested in transcription factors because they help confer upon cells their cell identity. And of course, we're interested in axon guidance genes because they help, in principle, they can help axons get from the olfactory epithelium to the appropriate location in the olfactory bulb. Let me just give you a few examples of the genes that we identified, just to give you a flavor of what the actual data look like. See? Great. So here, we're going to look in this quadrant at the expression of 3 transcription factors that belong to the NFI family, which I'll bring up later. And these are transcription factors that are known to be important for olfactory sensory neuron development. And you can see all 3 of them are expressed as gradients that vary as a function of each OSN subtype's DV score. NQ01 is that marker protein that was previously thought to uniformly mark the dorsal domain of the epithelium. And what you see is that this marker is not expressed in cells that have a ventral DV score. It's not expressed in low DV scores. But once cells become dorsal, it is expressed. And interestingly enough, it's not expressed uniformly. It's expressed in a gradient. Here are the 2 axon guidance genes that were known to help guide a zone in the nose to the right zone in the bulb, neuropilin-2 and robo-2. And you see these are both themselves organized as gradients as a function of the DV score. So the presence of all these gradients, together with the idea that maybe the JEP dorsal and JEP ventral are conveying some information about dorsality or ventrality, suggested to us that maybe there aren't 4 zones. Maybe instead, each neuron has a unique DV score, and that DV score represents where it is in the epithelium going dorsal to ventral. Okay? If that's true, instead of having 4 zones, because each olfactory sensory neuron, on average, has a unique DV score associated with a particular receptor that was chosen, you would imagine basically 1,000 zones or 1,000 peaks of expression going from dorsal to ventral. Is that true? So we decided to explore that possibility via the use of the technique MERFISH, which allows us to do essentially in situ analysis for hundreds of odor receptor genes and hundreds of DV genes and markers all at once in parallel. Here's a coronal section of the olfactory epithelium. Here we're marking mature olfactory sensory neurons and stem cells in green. You can see them, support cells in red, and cells that belong to the lamina propria, which I'm going to get back to, here in blue. So this technique works. Here you're looking at in situ via MERFISH of all those cells that express one particular odor receptor. I just want to mention that, of course, the actual cells are very small, and so we've blown up these dots 100 times. But you can see kind of a spatial distribution for this particular ventral receptor that can be easily detected by MERFISH. Okay, so we have in situ data for ORs. We have in situ data for the DV score. Does the DV score, in fact, describe an axis that runs from dorsal to ventral? And are odor receptors actually building a map that's running from dorsal to ventral? So this is what we see. So on the left here, we're going to take each mature olfactory sensory neuron and color it by its DV score. And what you see is this beautiful gradient running medial to distal, which effectively is dorsal to ventral, going from here to here, from red to blue, along each one of these little turbinates. And that tells you that each cell in the nose has a unique DV score, and that DV score corresponds directly to the cell's spatial position going dorsal to ventral, top to bottom in the epithelium. Is there actually a map built out of photoreceptors? In this plot, and by the way, here we've just kind of taken out some of these like little curlicues and zoomed in so you can really beautifully see these gradients, okay, these spatial gradients. Is there actually a map of photoreceptors? Here, we've identified the photoreceptor expressed by each neuron, and we've looked up what the associated DV score is. And again, we see this beautiful gradient going dorsal to ventral along all of these little curlicues that make up the turbinates. And the fact that we see these continuous gradients taken with the fact that every OSN subtype is associated with a unique DV score means that there are 1,000 different peaks of odor receptor expression on average that are organized into a map. Okay, so there's a map of odor receptor expression in the nose. There aren't 4 zones. There are 1,000 of these zones where the position of each odor receptor varies continuously, from receptor 0 here to receptor 1,000 over here. And if you just want to see these distributions a little bit more to convince yourselves, here you're looking at the cumulative distribution in space of the expression of each of these receptors. And if you look at these 2 receptors that have very similar DV scores, and you ask what are their spatial positions, you see they have highly overlapping distributions, which is to be expected, but the means of these distributions are clearly distinguishable. And the fact that the means of these distributions are clearly distinguishable means that we can make accurate predictions based upon an individual cell's DV score of where it's going to be in the epithelium. And if you give me a spot in the epithelium, I can make the other prediction. I can tell you what its DV score is going to be. Again, consistent with the idea that there is some sort of underlying map. I showed you in situs previously, and we talked about this idea that there's zones, which suggested that actually the distribution of each receptor is broad. How can there be a map of 1,000 things if they're so overlapping? Here we're looking at the distribution of DV scores for each OSN subtype on average, both in our single-cell sequencing data and in our MERFISH data. And you see that in both cases there's this sharp peak, which corresponds to each odorant receptor having a sharp peak of expression in the epithelium. But there is a distribution. And I think this distribution explains why previously researchers believed that there were zones. I think, so one thing that we have the ability to do, because we have so much data, is we can ask whether or not the rank order of odorant receptor expression along the dorsoventral axis of the olfactory epithelium is shared across individuals. If this is really a map, the map in my nose should be the same as the map in your nose. And what we find from these data is that there's a 0.99 correlation in the rank order of odorant receptors going dorsal to ventral. So the map that we are observing is incredibly precise and conserved across individuals, making it a real map. And so this raises, the broad set of questions. What is the relationship between space, the DV score, and OR expression? Everything I've shown you suggests that all 3 of these things are related. Almost certainly that the DV score represents position in space. But there must be some relationship between the space and DV score and OR expression in some way, because it's clear that ORs are organized into this patterned 1,000 different stripes from dorsal to ventral. So what might establish kind of these relationships? One naive idea is that maybe activity coming downstream of each receptor, which would be expected to be unique in a given odor environment, maybe that's determining where neurons are ending up in space, or maybe it's determining the DV score. And that turns out absolutely not to be true. So if we do some simple manipulations, like say we just close one nostril, right? If you do that, you deny 1/2 of the nose any odor stimulation. We can then sequence that side of the nose and compare it to the nostril that was open, and we see that DV scores don't change at all. We can make even more sophisticated manipulations. We can generate a mouse with a monoclonal nose that only largely expresses in 95% of its neurons one odor receptor, and this receptor is splayed out over the entire epithelium. If the OR 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 activity is determining DV scores, you'd expect all of those cells to have the same DV score, except instead, as you see here in blue, they have a complete diversity of DV scores, suggesting that DV scores represent where you are in space, and they aren't determined in any way by the odor receptor that's being expressed or neural activity. Okay, so if that's true, what you might imagine, like a model you might build in your head, is that, well, then maybe space determines the DV scores. Maybe in some way DV scores determine which OR is expressed. If that idea is true, if that model is true, if we're going to go from space to DV score to ORs, then you would expect that DV score genes, some of those 300 genes, should not just be expressed in mature olfactory sensory neurons, but they should also be expressed in precursors before ORs are chosen. And maybe the DV score genes guide the choice based upon space. So we can begin to think about this because, again, in our data set, we have 400,000 olfactory stem cells and precursors. So here we're going to isolate just those 400,000 cells, and we're going to exclude all the mature olfactory sensory neurons. And here you can see kind of a cartoon of the different intermediate stages. And we're going to embed all of those transcriptomes, again, into a UMAP plot. So each dot here is a cell. And we're going to color each one of these cells by what's called pseudotime, which reflects the developmental trajectory these transcriptomes take from stem cells to near fully developed olfactory sensory neurons. And if we look at marker genes that are known to be associated with these stages, we find that they're reliably kind of expressed along this axis. So this axis really does reflect developmental time. So these cells are stem cells. These cells are about to be mature. And you can see that also because they are just beginning to express odor receptors. All right. So do these precursors express DV genes? So the answer turns out to be yes. So here we're going to illustrate the expression of DV genes on this very same UMAP plot. You see, even at the stem cell level, some stem cells have high DV scores, others have low DV scores. And very early on, gradients of the DV score begin to be established, such that here there's some cells with very high DV scores, some cells with intermediate DV scores, and some cells with low DV scores. And so even before the odor receptor is ever chosen, cells know what their DV score is, and therefore they must know where they are in physical space. Importantly, if we just look at our Merfish data, and we kind of look at DV scores in that stem cell layer underneath the mature olfactory sensory layer, we actually also see these nice gradients in DV scores, consistent with the idea that stem cells know where they are in space. Okay, so we know that space is determining DV scores, and that's somehow influencing odor receptor choice. You might then have the idea that the stem cells know where they are in space, and therefore are somehow fated to generate a particular odor receptor. Okay, there might be like a fate relationship where like each stem cell is assigned to a particular receptor, and that's how you get 1,000 receptors precisely ordered from dorsal to ventral. So we wanted to explore that idea, and so in order to explore it, we performed the first formal comprehensive fate mapping ever performed in the olfactory system. So here, in order to do this, we took an animal with its nose, and we gave it a drug that killed all of the mature sensory neurons, leaving only the stem cells below. We then infected the nose with a bar-coded lentivirus that infects stem cells and permanently labels their DNA with a DNA barcode so we can identify them in single-cell sequencing data. We then waited a month or so, and these stem cells, of course, divided and rebuilt the epithelium from scratch. They generated new precursors and new mature olfactory sensory neurons, as you can see here. And then we can take these recovered noses, break them up, do single-cell sequencing, and ask about the clonal relationships between each stem cell and the mature OSN progeny it generated. And one thing you might imagine is that maybe each stem cell only gives rise to one type of OSN. Maybe it only gives rise to, each stem cell is fated to pick a particular receptor. That turns out not to be true. Instead, each stem cell seems to pick a very restricted set of receptors that hover around the mean DV score associated with that stem cell. And so here you see the DV scores of all of the progeny associated with the different clones that we have. And you see they all have a kind of mean DV score that has a very narrow band. Okay? And if we look at the distribution of these DV scores, we see that there's a very sharp peak with a little bit of a distribution around it. What this means practically is that each olfactory stem cell has an ideal favorite type of receptor it wants to pick. And it very frequently picks that receptor. But often it makes mistakes because of biology, which explains this distribution, and also explains why there's a kind of spatial distribution of each receptor along the dorsiventral axis. Okay, so here I'm describing to you, how it is that space might influence odorant receptor choice. But what I haven't told you anything about is how it is that the stem cells know anything about space. So how do they know? And so we were guided by work that others had done and by some interesting properties of our data set, which suggested that maybe the way that space is encoded in the epithelium is through a gradient of retinoic acid. And we think that because there's this enzyme, ALDH1A2, that is expressed in the lamina propria underneath the olfactory epithelium. And this enzyme, as you can see here in our Murphy's data, is expressed in a dorsal to ventral gradient, suggesting that there is a gradient of retinoic acid running from along the dorsal ventral axis of the olfactory epithelium. And interestingly, if we look at the genes that belong to the DV score, we see a ton of genes that are all organized in dorsoventral gradients that seem to either be involved in retinoic acid metabolism or in binding to and responding to retinoic acid, suggesting that retinoic acid might be a master controller that tells stem cells and progeny where they are in the epithelium, where they are in space, right? Just like I told you was happening in the retina. So we can test this by either adding to the nose a retinoic acid inhibitor, thereby blocking retinoic acid signaling, or just adding all-trans retinoic acid to the epithelium. And if we do that, we find that we can actually shift the position of receptors along the dorsoventral axis. So if we inhibit retinoic acid, now what we see is that there's a dorsalization of the epithelium. That is, the epithelium tends to generate cells with more dorsal DV scores, and it tends to pick more dorsal receptors, as you can see here, and it tends to generate fewer ventral cells and tends to pick fewer ventral receptors. Conversely, if we add extra retinoic acid, we see the opposite pattern. In this case, the epithelium is ventralized. It tends to pick more ventral receptors and express more ventral DV scores. It tends to de-express dorsal receptors, and it tends to have fewer dorsal DV scores amongst its cells. In other words, by manipulating retinoic acid up and down, I can shift this map of 1,000 receptors up or down in space. Okay, and importantly, the shift that we're seeing up and down in space is not only apparent in mature olfactory sensory neurons, but as you see here, it's also apparent in stem cells. And so it seems like retinoic acid is a key signaling molecule that tells stem cells and mature olfactory sensory neurons where they are in space and kicks off some sort of cascade that ensures that the correct receptor is chosen at the right place in the epithelium. Okay, so how might that process actually occur? Well, we hypothesize that maybe the promoters of each OR gene contain binding sites for transcription factors that are sensitive to these gradients that we're describing. And so here we're doing an analysis of the number of binding sites for particular transcription factors. I'm not going to discuss these two for now, although they're important transcription factors for olfactory epithelial development. But notice these two transcription factors. You can see that the transcription factor RAR, which is a retinoic acid responsive transcription factor, has an increasing number of binding sites as cells become more and more ventral. And I told you that retinoic acid, the ligand for this thing, also is expressed in a dorsal to ventral gradient. So you might imagine that this kind of pattern would allow individual odor receptors to be organized along the dorsal ventral axis. Similarly, those NFI transcription factors that I showed you at the very beginning of this talk, that themselves are organized in gradients, binding sites for NFI are also enriched in more ventral receptors compared to more dorsal receptors. So all combined, you can imagine that these differences in the transcription factor binding sites in front of odorant receptors, taken with all these gradients that I'm describing, could build these 1,000 different stripes. And I'll tell you, but I won't show you, that we've proved that in a number of ways. One simple way you can do it is by making a genetically modified mouse and taking the promoter of one odorant receptor gene and putting it in front of the actual gene for a different odorant receptor gene. That actually moves the receptor in a predictable way in space, consistent with the idea that the genome itself determines where in the epithelium each olfactory sensory neuron is going to be expressed. Okay, so this describes a little bit to you what we think is going on with the transcription factors that are part of the DV score. Remember there's that other class of genes that I talked about? I told you that there are 30 axon guidance genes, all of which are expressed in gradients in the olfactory epithelium. And so you might imagine that because there's so many of these axon guidance genes, that they might be sufficient to enable an olfactory sensory neuron in the epithelium based upon its position to find its correct glomerular target in the olfactory bulb. In other words, the existence of the map in the nose might effectively entail the existence of a map in the bulb. We wanted to test that idea, and so we took advantage of a data set that was recently developed, a spatial transcriptomics data set by my friend and colleague Alex Fleischman at Brown, which you can see here a little bit on the left, where he used spatial transcriptomics to identify every receptor in the olfactory bulb. So spatially now in the mouse, we know the molecular identity of the receptor that innervates every single receptor in the olfactory bulb. Given that, we can then ask in the olfactory bulb itself whether or not the level of DV score gene expression by the innervating sensory neurons has any pattern in the bulb. And one thing I want to just mention, which I kind of glossed over at the beginning, is that each bulb is actually mirror symmetric and contains within it 2 hemibulbs. So I'm going to show you 2 hemibulbs here. And what you can see is that if you color each glomerulus in by the DV score of its innervating OSNs, you see that it forms this beautiful gradients. So you're not seeing zones, you're seeing these gorgeous gradients, suggesting that the genes that belong to the DV score contain within them enough information to actually guide an axon to its appropriate dorsiventral location in the olfactory bulb. And we can test that idea computationally. Indeed, DV scores do a great job of predicting the DV score that would be imputed by spatial position in the olfactory bulb. And I think importantly, if we just use the DV score to predict the actual 3D position of glomeruli around the entire glomerulus, we can do so very accurately with only a 400 micron error. To give you a sense of scale, each glomerulus is about 150 microns big. And so what this means is that with just this single number, I can predict the 3D location of glomeruli with like a 1 to 2 glomerulus error in a structure that contains 2,000 glomeruli and is several millimeters big. And so there's a huge amount of information in this DV score about the actual 3D location that axons go to in the bulb, suggesting that the map in the nose really can entail the map in the bulb. Okay, last slide. I told you that key sensory features are organized into maps in the retina and in the cochlea. And so you might imagine that key chemical features are spatially organized in the nose. This is an old idea called chematopy. You might imagine the receptors that respond to similar chemicals might be localized to particular domains in the olfactory epithelium. And maybe the reason why there's this highly precise map is to support the generation of a chemical map in your nose. Is that true? So my lab has developed a technique called ACT-seq that allows us for any odor to identify all of the receptors in the nose that respond to that odor. I'm not going to show you any of those data. Just know that for a whole bunch of odors, for 22, for 50, maybe even at this point 100 odors, I know the complete set of receptors in vivo that respond to those odors. I can then go to the olfactory epithelia merfish data that I showed you before and just color in the neurons and their position in space that are responsive to any given odor. Okay? And so you see basically 2 things in these data. The first is that acids, like octanoic acid or nonanoic acid, tend to activate only dorsal cells. And that's a thing that's well known in the olfactory literature. There's an enrichment, for some reason, for acid-responsive receptors in the dorsal domain. And I will just say, for those of you who are really interested in olfaction, that we think the dorsal domain actually is the one real zone in the bulb. It seems to be special, and it seems to be somehow enriched for a particular type of receptor. But for all of these other generic odorants, basically they activate all of the ventral domain. Right? It's just totally intermingled. And if you look at these spatial patterns, there's no way to distinguish whether or not an animal is smelling octanone versus hexyl acetate. And that is very different from what we can do mathematically if you tell me the identity of the odorant receptors that each of these odors is activating. If you tell me the identity with 100% accuracy, I can tell these odors apart. Okay, so there is a clear spatial map of odorant receptors in the nose, but it does not organize chemistry. Okay, so let me just summarize what I told you. We started with a discussion of kind of where we were in olfaction, and I described to you this idea that basically odorant receptor choice is random, that there's disorder in the nose, and somehow magically order appears in the olfactory bulb. And what I've shown you today is that the nose is just like the other peripheral sensory organs. At long last, we have discovered a peripheral sensory map for olfaction, and this map organizes along the dorsiventral axis of the olfactory epithelium all 1,000 odorant receptors into basically peaks that run from the top to the bottom in a prescribed order in a manner that's conserved from animal to animal to animal. Okay? And that, I would argue, means that the nose is the most patterned nervous tissue in your entire body, okay, which I think is rather remarkable. And I'll say that part of the reason, if you read the literature, that people for so long had hewed to the idea that receptors must be chosen randomly is because no one could imagine that development could solve the problem of putting 1,000 things in precise places. And what I show here is that, in fact, it can. So why build this map? As I just described, it's not to organize chemical space. We think there's two likely reasons. One reason is for robustness. Chemical tuning of individual receptors are typically very broad and often very redundant. And so by kind of tiling the nose with all these different receptors, if you injure one part of the nose, you'll still be able to smell most of odor space with the remainder of the nose, which is something that's not possible if the receptors are organizing chemical space. So we think this is a mechanism for robustness. We also think that this is probably important because it allows spatial position in the nose to determine the spatial position of glomeruli in the bulb. In other words, through one program in which retinoic acid determines DV genes, you build two maps in parallel. So it allows development to solve two really hard problems just once. I'll mention that I think this work has substantial implications for therapeutics. There are millions of Americans that have lost their sense of smell. We're really interested in building prostheses for them or using stem cells for them. And understanding this basic physiology is essential if we're ever going to fix people with broken noses. And I think the precision of the mechanisms that we've described really suggests that subtle deviations and gradients during development across the entire brain might actually have profound functional consequences. So I think it changes the way we think a little bit about neural development. Okay, so all of the work that I've shown you today was done by this guy, David Brand, who's a graduate student in my lab. He's a brilliant graduate student, along with some help by Tetsuya Sukahara, who's a former postdoc in my lab. And some work is a collaboration between my lab and Tom Bose's lab at Northwestern. So now is normally the time when I thank the NIH for its generous support of all the work that we've done, and I am truly grateful. For many, many years, I've been allowed to be a steward of public funds to do exactly this kind of exploratory, data-driven research, and I think it's taught us a lot, and I'm really proud of what we've accomplished. I think as many as you know, all of the funding at Harvard has been terminated. We haven't any funding anymore to support any of this work. I think there are pervasively challenging things about that. I just want to briefly focus on the consequences for the trainees. I think we're facing an unprecedented amount of uncertainty. David is a genius. And if you talk to David about why he does science, he'll tell you that he does it because he's curious, because he wants to know the answer, because he wants to figure out how things work. But ultimately, he'll tell you that he does it because he feels like it's a way that he can contribute to this country. He feels like in doing science and adding to the literature, he's helping us be the best Americans we can be. And he views science fundamentally as a service, a view that I very much share. And it's a view that I know all of you share. I mean, all of you here at the NIH, you're involved in cutting-edge research and medical care. But truly, I think all of us appreciate that all of you are doing science as a service. And so speaking from like my seat at Harvard, we know that there's a lot of stuff going on at Harvard, and we know there's a lot of stuff going on here. And I just want you all to know we see you, and we're super grateful for everything you've done, both on the intramural side and extramural side. We see your service, and all of us appreciate it. So thank you very much, all of you, for everything that all of you are doing. And I appreciate your attention, and I'll take any questions you might have.
Thank you. People here certainly know where the mics are, and we've got some questions coming. No, we don't. Okay. But I do have a few online. I can't quite see behind me. Okay, we'll start in here. Go ahead. Yeah.
Thank you very much. It's a wonderful talk. So kind of question is like retinoic acid looks like master regulator of many things. Do you have a map of lamina propria cells that produce retinoic acid? Does it really has some gradients, or what type of cells making it? And
yeah, so we think that mesenchymal cells in lamina propria, and that's as far as we've got, contain a metabolic enzyme that converts retinaldehyde to retinoic acid. And so that allows us to infer that there's going to be this gradient. What would be amazing is if we could directly visualize the retinoic acid gradient. Of course, there's tools to do that, and that's a thing that we're doing now. But what I think is super interesting about this, let's just talk about it for a second, is this is an example where basically the cartilage is patterning the nervous system. So just think about that for a second. That's not how we're used to thinking about it, but here literally that's what's going on. So it's also very unusual and very interesting, and it's obviously a thing we're interested in working on. Thank you for your question.
Thank you very much.
Thank you so much for the talk. It was beautiful. I have two questions. You can pick so that we can keep the line going. So I was wondering first, I understand that you kind of have developmental time even in the adult here because you have this continuously regenerating. I'm wondering if you know a little bit about how this map is established in development in terms of the animal's age. And my other question is whether there might be other there don't have to be functional maps, but whether either valence or action selection or source might be other kinds of maps that might be there.
I'm going to answer both those questions. It's okay?
Yeah.
All right. So the first question was, I keep talking about the adult as if it's development, but it's not development. Of course, the map originally develops in an embryo, and then it's regenerated in the adult. And so I kind of leverage the adults to think about the ontogeny of the map. It turns out we know, not a lot, but we know something about how things are organized in the embryo to build things originally. And I think it's very likely that different molecular determinants originally build the map. And that the map, this retinoic acid mechanism I'm describing, probably isn't sufficient to explain the embryogenesis of this structure. There are probably many, many other determinants and gradients that are involved in that. But it probably is sufficient to maintain the adult tissue. So that's the first question. The second question you asked was, I've looked at chemical space, right? And I kind of see this very weak dorsal to ventral differentiation. But there's many other axes along which odors can be organized. Is it possible that those are organized in space? My answer to your question is yes, that's absolutely possible, and we're looking at that now. We're continually generating these AXIC data for different categories of odorants, and you might imagine that there is some spatial organization to where they are in the epithelium. One thing I just want to say, just to like full disclosure, so we're all on the same page here, is that of course the set of receptors that we're going to identify as being active depends on the concentration of the odor we use. So the degree to which a map will be apparent may or may not depend on the actual concentrations we're testing. And so we're beginning to do this now across a range of concentrations. It's not like we see a map anyway, but we're aware that that's a limitation of what I showed you, and we're thinking about it carefully. So thank you for your question. Josh?
Thank you, Bob. This was an inspiring talk. I'm really, really pleased to share the time with you. Follow-up, similar question, but more of a clinical bend. I'm curious, and I'm confident that at best you'll be able to share a hypothesis, but I'm very curious as to what it is. In the established model and following injury, thinking our patients with parosmia, other qualitative olfactory disorders, do you believe that there may be an association with disruption of this map related to their symptoms?
Yeah, I think that's a really wonderful question. I think the answer is yes. In many cases, basically stem cell biology gets changed here a little bit. And so it could recenter our attention both on understanding and restoring that stem cell biology to build an appropriate map. As I told you, it's almost certain that overall perception depends on patterns of activity in the bulb. And as I told you, this one gene expression program that's pervasive in stem cells and precursors coordinates space in the nose and targeting in the bulb. And so the stem cell biology here is really critical, and I think important to focus on for therapeutics. So thank you for your question.
Thank you.
I wonder whether the causation might work a little differently from the way you're envisioning it. Is it possible that the glomerulus that a cell reaches is not precisely determined, but which one it reaches determines to some extent which odorant receptor will express and other features of the cell?
It's a great question. So one might imagine that you build this targeting, and somehow there's a retrograde signal that comes back and tells the cell which odor receptor to express. So I'm sorry I wasn't clear about this. It turns out that receptors are expressed way before the axons reach their target, and in fact, people have long theorized that the receptor itself somehow governs the expression of axon guidance genes, or governs guidance in some way to get axons to the right target. We show that it's largely not true, right, in these data, but that idea derived from the fact that the receptor is chosen very early, well before the axon is actually elaborated. But your idea is an idea that many have had, so thank you for that question. It's a good question.
I have a lot of questions. I'll ask two. So one is the program that you're suggesting, particularly the dependence on retinoic acid for dorsiventral patterning, is very similar, it sounds, to patterning, for example, in the otocyst, which is the NRER precursor. And I was wondering, to what extent have you looked across other systems in the body that have dorsal ventral patterning, and have you looked for commonalities versus differences?
That is a wonderful question. My friend and colleague Lisa Goodrich has a very large single-cell sequencing data set of many of the cell types in the ear, and we're beginning to think about that. The other place where there are actually interesting similarities in terms of overall patterns of gene expression are actually in DRG neurons. So we've been talking to David Ginty about that. But I don't have anything to say about it. But I think this is like, I think there are common principles here, obviously, that I think really matter. So yeah, we're thinking very much about the comparative mechanisms as well.
Okay. So my second question, I'm an ENT, and traditional teaching is that the olfactory sensory epithelium is really, in human, located more in the rostral area, right, of the nose.
Yeah.
Yep. So how does this map correlate, right, with the human sensory epithelium?
Yeah, so we've been collaborating with Brett Goldstein at Duke to do two types of experiments, both of which are really fun. So the first is we're assembling, and many others are doing this, but we also are doing it. We're assembling an atlas of human olfactory sensory neurons. We now have many thousands of neurons in that atlas. And you'd expect to see, for example, gradients of dorsal ventral genes in the human or not, depending on the way the anatomy is organized. And we're just getting into that analysis now. I will tell you, though, about like a fun experiment we're doing, which I'm like super excited about, just because it's fun. I think you'll like it. We're doing the first real molecular biology of the nervous system in living humans. And so what I mean by that is, in the mouse, I didn't tell you how we did it, but I told you that we have this method called AXSeq that lets us know which neurons are activated for a given smell. The way that works is you take a mouse, you give it an odor, you kill it 5 minutes later, 30 minutes later, you sequence its nose, and we have identified a 200-gene signature that reflects the acute activation of an olfactory sensory neuron. Okay? So we can synthesize that into a score, and that lets us identify all the receptors that are activated and how much they're activated across the entire repertoire. Turns out that works in humans. And so we've been giving smells to patients, let them smell like lemon or orange or whatever, pizza, and then we let them smell for 30 minutes. We then take a biopsy of their nose, and we sequence all the cells, and we can get the same molecular signatures back. This proves, by the way, that the immediate early gene FOS, which has been proposed as this ubiquitous marker of activity by Mike Greenberg in 1984, actually is recruited by neural activity in humans because we can see it in a living human in response to a real cue. And we think that this could be a useful way of thinking about using the nose as a biomarker for all sorts of things. Right? It's affected in psychiatric disorders, potentially. It's certainly affected in neurodegenerative disorders. It's sensitive to immunity. And so it could be like a neat way of really getting at the underlying molecular variation that occurs during various disease states. And so we're beginning to think about that as well.
Thank you.
Of course.
Hi, great talk. I was thinking about your transcription factor binding site data. I noticed that you only looked at 750 to 1,000 base pairs before upstream of the transcription start site. I was wondering if you have any sort of chromatin looping data to
Yeah, so my friend and colleague Stavros Lomvardis at Columbia is the world's expert on looking at 3D genome architecture with respect to odor and receptor genes during development in immature olfactory sensory neurons. And the reason we focus on that window is because Stavros thinks that most of the action happens in that little window. It's possible there are enhancers that are upstream, but basically from his data, guided by his work, we think that pretty much things are pretty close by the OR. And that would kind of make sense, let's remind you, from an evolutionary perspective. So these are the fastest evolving genes in your genome, well, close to. They're almost as fast evolving as genes that are related to the immune system. And they evolve through duplication, right, and then variation. And so, you kind of want, if you're going to duplicate gene families rapidly, you kind of want things to be close by so that you can copy them and paste them and kind of keep regulatory elements close by so that things aren't broken frequently by evolution. And that seems to be the pressure that guides this particular architecture.
Okay, great. Thanks.
We do have can I ask a couple questions from online? I'm sorry. It's been building up. There is a question about the Alzheimer's disease and COVID connection to anosmia. Mm-hmm. What can you say about that?
Sure. So I'll just say that we were accumulating this very large data set of 5.5 million cells when COVID broke out. And because we had a subset of the data, at that point, 500,000 of these cells, plus we had the human data that I described earlier, we were able to look through those data and come up with a hypothesis for how COVID might affect the sense of smell, which we believe, and I think subsequent studies in humans and in animals have verified this, we believe occurs because the virus attacks the support cells that I mentioned, but not the neurons directly. And that in part explains why people tend to recover their sense of smell rather quickly, because the nervous system is largely intact, but the support cells have been infected. But then when the infection's cleared, the whole system can recover quite quickly. And for Alzheimer's, I would say that we still don't understand the level at which Alzheimer's affects the olfactory system. It's clearly a harbinger of, if you lose your sense of smell, it's clearly a harbinger of future, potential for neurodegenerative disease. I think your relative risk is like 10 times if you have, MCI and you suddenly lose your sense of smell. My dad has Parkinson's. He was anosmic 10 years before he developed full-blown Parkinson's symptoms. It's very common. Okay, we don't really understand these interrelationships, but it probably has something to do with the fact that the smell centers are in the temporal lobe and associated with memory centers. And so I think people are thinking about this relationship, but I would say we don't fully understand it.
Okay. Maybe one more up there and then one more back online.
Hi.
Hi.
So I was just thinking that there are some neurodevelopmental disorders characterized by faulty migration of stem cells. And I think it would be really interesting to look at if that's something that occurs when even adulthood in those individuals with neurodevelopmental disorders, and that if that'll impact the migration and olfactory abilities and receptor expression on that dorsal ventral axis.
Yeah, we actually, a thing we don't understand is there's these stem cells that generate neurons, and we know the stem cells are below and the neurons are above. We don't really understand how far they migrate and the ways in which they actually the ways in which maybe like a receptor generated by this stem cell might actually change where it is in space, and how much that actually introduces noise into the system versus how much the migration is precise. And I think your suggestion to think about that in the context of neurodevelopmental disorders is very interesting. I'll also mention one other thing that I think you might find interesting. I told you there were 7 gene expression programs, right? It turns out that 2 of them, which we've published on in a paper a couple of years ago, actually encode a program that adapts the cell to levels of ongoing neural activity. Okay? So I don't want to explain it in any detail, but one important thing about it is if you look at ASD models in which genes that are involved in ASD are mutated, we know those animals and patients are hypersensitive to sensory stimuli, and it turns out that adaptive program is broken in olfactory sensory neurons. And so we think a lot about how these gene expression programs we've identified might actually serve different functions in the cell and might relate to disease, and this is one example of that.
That's awesome. Thank you.
Okay, maybe close with this one question. Generally speaking, the importance of the kind of animal studies you do and what's possible and not possible with non-animal models that people want to transition to.
Yeah, thank you for that question. Mm-hmm. My lab does a lot of computational work and a lot of computational modeling of various aspects of the nervous system. That's a mainstay of what we do, actually. And I will say that at least for the nervous system, I would argue we are not at a place where we can substitute animal research for in silico models or organoids. None of the things that I talked about today, none of these insights would be possible without actual access to in vivo models. We simply don't have the data. I think if you think about the brain more broadly, we don't have the training data yet to really build sophisticated models of nervous system function. I think everyone who's working to build those kinds of models would agree with that perspective. And I also think that brains are embodied, right? I think a lot of, as I mentioned at the beginning, I think a lot about the relationship between brain and natural behavior out in the world. And a lot of what the brain evolved to do and what your brains are doing now is helping you navigate a world in which you have a body that's real. And separating that body away from the brain, I think, from a research perspective, is a big mistake. And so I would argue that while it's an ideal goal to move to a place where we're in silico, and I can imagine in many domains, toxicity, maybe cancers at this point, you could begin to do that. I think for neuroscience, we're still at a stage where we should be investing heavily in animal research while still encouraging people to think about in silico solutions for scientific problems.
Okay, excellent. Comments like phenomenal talk, absolutely fascinating, excellent. So I'll leave you with that. And there's a reception out there, and let's thank the speaker.
Thank you all for your attention.